So we are going to start off our notes going over DNA. So remember that DNA stands for deoxyribonucleic acid. DNA is a nucleic acid that stores your genetic information. And the monomer of nucleic acids are nucleotides. And remember that there's three parts of a nucleotide, and you can see that in the picture here. There's a phosphate, deoxyribose is the sugar, and then the base is either going to be the A, G, C, or T. So the adenine, the thymine, the guanine, or the cytosine. And remember that when the two sides of the DNA are matched up, C always goes with G and A always goes with T. That's Chargaff's rule. When DNA needs to make a copy of itself, so remember this is happening in interphase in the S phase, the original strands of DNA open up, and then the new monomers, the new nucleotides, can get added on, and they just follow the base pairing rules. And DNA replication is said to be semi-conservative. DNA, in addition to just making copies of itself, DNA is also used as the code to make proteins. So remember we talked about how DNA inside of the nucleus can be rewritten or transcribed into mRNA, and then that mRNA message can leave the nucleus. It will go to a ribosome where it will be translated into a protein. And then proteins can just do all sorts of different things. They can serve as channels in the cell membrane. They can do cell communication. It could be an enzyme that just got made, so that could be used to speed up a reaction. But this is how all proteins are made, and it starts off with that genetic code that we call DNA. Something that we need to make sure we know is the differences between DNA and RNA. So they are both nucleic acids, which means they both have the same monomer of a nucleotide. So they both have a sugar, a base, and a phosphate. But the sugar is going to be different. So DNA is made out of deoxyribose sugar. RNA is ribose sugar. When it comes to the bases, they both have adenine, they both have guanine, and they both have cytosine. It's only that fourth one that's different. So DNA has that T that we call thymine, and RNA has the U that we call uracil. So there's never any T's in RNA. The final difference here is the number of strands. So DNA is always going to be double-stranded. There's two sides, just like a ladder, with the bases on the inside. RNA, though, is just half of a ladder, so we say it's single-stranded. Usually, our cells do a very good job of making sure that DNA is looking the way it's supposed to. But sometimes certain chemicals, or if you are in the sun for way too long over a number of years, mutations can't occur. It says here changes, so mutations are changes in the DNA. They can be harmful, beneficial, or neutral. So remember that mutations are the source of all new traits. So during the process of meiosis, when organisms are making egg and sperm cells, if a mutation occurs during this time, that mutation in either the egg or sperm cell or both will be passed on to the next generation. And then if that mutation was a good thing, if it was beneficial to the organism, then through natural selection, there is a chance that that new trait that started off as a mutation can now be passed on through that population. Okay, next we have genetics. So this is the study of genes, genetic variation, and heredity. So we started off by talking about just the simple genetics, which is the Mendelian genetics. So remember Gregor Mendel? He was that cool Austrian monk that studied the pea plants. And he gave us a lot of good information about genotypes and phenotypes. And he said that genotypes are just the genes an organism has. And they can come in different forms, and these are called alleles. He also realized by looking at the pea plants that there's dominant traits or there's dominant alleles that overpower the other allele every time. And there's also recessive alleles that can be hidden in an organism if they do have one dominant allele. We also need to make sure we have a very good understanding of these next three words here that are in light blue. So we have homozygous dominant, big T, heterozygous, big T, little t, and homozygous recessive, little t. Okay, so this is all information about genotype. We also have phenotype, which is the actual traits you see with your eyeballs. So examples, tall, red, four petals, short, hairy, round, whatever you can think of that you can see with your eyes, these are going to be phenotypes. Okay, I know that we did not forget the hundreds of Punnett squares that we did together because you all just loved doing these. I know you did. So Punnett squares are used to make predictions about what the genotypes are going to be of the offspring. And then once you can make predictions of the genotypes, then you can use that to figure out what the offspring might look like. So you can figure out their phenotype. Okay, so this sample question I have here is how many offspring in this big dihybrid cross can be expected to be gray and small? So if you check out this dihybrid cross here, I even put the word egg and sperm on the outside here. So you can remember that the gametes from the parents go on the outside of these Punnett squares. So it's very important to remember that. So when you look, so let's just look at the sperm category at the top here. You will see there's 4 different possible gametes for this particular organism. They're all the same. It's going to be recessive for both alleles, but there's only ever going to be one G and one B in each gamete, ever. It never doubles up. There's one allele per trait in a gamete, and that is because the gametes are haploid, not diploid. That is very important. Okay, so going back to our question here, how many will be gray and small? Well, it looks like gray is the dominant trait with the big G's, and then the little b is small. When I look in this dihybrid cross here, I see that 4 out of 16, or you can reduce that to 1 out of 4, follow this criteria here of being gray and small. Okay, some other things we talked about that do not follow Mendel's rules are incomplete dominance versus codominance. So incomplete dominance is this example on the left here, where you have 2 different colors of this fish. So we have red alleles and we have blue alleles. And they both show as red and blue in the homozygous genotypes. But when you have that heterozygous genotype, both alleles show together. Okay, we also have over here on the right side incomplete dominance. And this one, that heterozygous individual shows the blended color or the blended shape or whatever you're looking at, it's a blend. Or you could say it's an intermediate or middle phenotype. Okay, something else we talked about was the sex-linked traits. Usually these traits are going to be found on the X chromosome, but they could also be on the Y chromosome. Okay, so just to refresh our brains on this, I have a sample question here. So it says, a man who has an X-linked recessive disease called hemophilia plans to have children with a normal female. What is the chance they will have a child with hemophilia? So if we look at the Punnett square over here, we'll see the female on top, and she is normal, not a carrier, so she is homozygous dominant. The male is on the left side over here. He has hemophilia, which means he has the recessive disorder. So since this is an X-linked disorder, we will put that recessive allele on that X chromosome. And so back to our question here. What is the chance they will have a child with hemophilia? So when I look at the inside of that Punnett square there, I see that the offspring that are XX, so those are the females, they are both carriers. So 100% of the girls will be carriers. And then the boys who are XY, they have a dominant allele. So they are perfectly fine. They are normal. They're not carriers. They don't have hemophilia. They're 100% normal. Okay, last thing here. So